All right, let's talk about one of the most basic ideas in all of biology, the gene. Now, for the longest time, we all had this picture in our heads of a gene as a simple, straightforward instruction manual for life. But you know what? The real story is so much weirder, so much cleverer, and honestly, way more amazing than we ever gave it credit for.
So, right off the bat, let's chew on this little puzzle. Humans, with all our complexity, have about 25,000 genes. A tiny fruit fly, it's got 14,000. I mean, how does that even make sense? Some plants have way more genes than we do. This whole thing seems like a paradox, and today we're going to untangle it.
Okay, to figure this out, we've got to take a step back in time. We need to start with that classic, old school idea of the gene, probably the one you learned about in high school. Let's call this the age of the simple blueprint.
So, the big idea back then, cooked up by the legendary Francis Crick in 1958, was something called collinearity. And it sounds fancy, but it was beautifully simple. The sequence of letters in a gene's DNA lined up perfectly with the sequence of building blocks, the amino acids, in a protein. It was a straight line, one for one, simple as that.
Yeah, that neat and tidy picture, it was about to get blown to smithereens because scientists started to realize that the genetic blueprint wasn't a nice, continuous line of code at all. Nope, it was full of gaps. The first crack in the foundation was a numbers game. Scientists were looking at our DNA and going, wait a minute, there's way too much stuff here. We had way more DNA than we should have needed to build all of our proteins. At first, they just shrugged and called it junk DNA, but there was so much of it, it felt like a huge piece of the puzzle was missing. And then came the experiment that changed everything. It was just brilliant. Researchers took the DNA for a gene and mixed it with its messenger RNA. That's the little molecule that actually carries the instructions out to the protein factory. They let the two stick together, and then they looked under an electron microscope. And what they saw was shocking. Weird floppy loops of DNA just hanging out, not attached to the RNA at all. This was the smoking gun. This is basically what they saw. It was a total game changer. It meant that big chunks of the original gene were being edited out before the final message was sent. So science needed a whole new vocabulary. Those weird loops were named introns because they interrupt the code, and the parts that actually made it out the door, those were called exons because they're the parts that get expressed. The introns get snipped out, and the exons get stitched together. Wild, right? Now you might be thinking, cutting and pasting? That sounds kind of wasteful and inefficient. But it turns out, this is where the real magic happens. This is one of nature's greatest tricks, and it's the key to creating mind-boggling complexity from a pretty small number of genes. This incredible trick is called alternative splicing. See, the cell doesn't just cut out the introns and glue the exons back together the same way every time. Oh no. It can pick and choose which exons to include in the final message. It can shuffle the deck. So from one single gene, you can create a whole menu of different proteins. It's like a gene isn't just one recipe, it's a page in a cookbook with tons of optional ingredients you can mix and match. Let me give you a killer example, the calcitonin gene. In your thyroid gland, the cell splices together exons 1, 2, 3, and 4. The result? A hormone called calcitonin. But in your brain cells, the cell uses the exact same gene and splices it differently. It uses exons 1, 2, 3, 5, and 6. And what does it make? A totally different molecule called CGRP, which works in your brain. One gene, two completely different jobs, just based on how it's edited. And don't think this is just some weird one-off thing. We're talking about a whopping 60%. 60% of all human genes using this trick. This isn't the exception to the rule. This is the rule for how we build complexity. The potential here is just mind-blowing. Get this. Researchers found that a single gene in a fruit fly can be spliced in so many ways that it could potentially create around 38,000 different proteins. And that right there is the answer to our puzzle from the beginning. It's not about having more genes, it's about being smarter with the genes you have. Okay, but the story of all this extra DNA doesn't stop with introns. Because our genome isn't just a workshop, it's also a museum. And it's full of fossils, the ghosts of genes from our deep evolutionary past. These are called pseudogenes. Pseudo means false, so false genes. The name kind of says it all. They're these molecular fossils, basically broken copies of genes that stopped working millions of years ago. They look like a gene, they smell like a gene, but they've picked up so many mutations over time that they're completely non-functional. So how does a gene even become a fossil? Well, there are two main paths. The first way is pretty straightforward. A gene gets accidentally duplicated, and that extra copy is free to just collect typos and mutations until it eventually breaks. The second way, though, is super weird. An mRNA message gets copied backwards into DNA and then randomly jammed back into the genome. It's like a photocopy of a final email draft being glued back into the original document.
Okay, so who cares about broken genes, right? Well, they're like finding a Rosetta Stone for evolution. For instance, most mammals have a working gene called GLO that lets them make their own vitamin C. We can't. Neither can other primates like chimps and gorillas. And here's the kicker. We all share the exact same typos that broke that gene. That is incredible evidence that the gene broke just once in a common ancestor we all share millions of years ago. So you can see how after all this, after finding gaps in introns, alternative splicing, and these genetic fossils, that old, simple idea of a one-to-one gene blueprint was just left in the dust. It didn't work anymore. It was time for a rewrite. So let's try to pin this down. What is a gene really? Well, it's not just the part that codes for a protein. It's the whole shebang. It's the entire stretch of DNA that gets read into an RNA molecule, including the coding exons and the non-coding introns that get edited out. It's a region whose parts can be shuffled and remixed to create a whole host of different things. The modern idea of a gene is just so much more flexible and powerful than we ever thought. If you take one thing away from all this, let it be this. The gene is not a static blueprint. It's not a rigid set of instructions. It's more like a dynamic, modular toolkit, like a Lego set that life can use with incredible creativity to build all the amazing complexity we see around us from a surprisingly small number of pieces. And that leaves us with a final, kind of mind-bending thought. For decades, we dismissed huge portions of our DNA as junk. But now we know that this so-called junk contains fossils from our evolutionary past and even plays a role in controlling our active genes. It just makes you wonder, what other profound secrets are buried in the vast, uncharted territory of our DNA, just waiting for us to find them?